So saying there is a theory about this essentiality of the hubs, which means the theory is like centrality and lethality in each protein-protein interaction network. So what is centrality? Centrality is if, for example, centrality of a node is if this node is a hub or not. So it's a hub, so it's very central. And lethality is that it's given by the graph that I showed you, that these hubs have higher probability to be lethal to the organism. So this is the theory of centrality and lethality in each protein-protein interaction network, also from Barabasi. But this is not really, as I said to you, not every hub it's essential, right? And when you go to plants, this is even more complex because plants are sessile organisms, which means that it seems actually that we are not really sure about that the hubs, this lethality and centrality theory it's not really applied for plant networks because, for example, here we have an example in yellow are 4 hubs in which the mutant, when you remove this node, sometimes you have no phenotype. I mean, you have no effect on plant growth, for example. So this is something that we are going to investigate, but it seems like it's our hypothesis that the centrality and lethality theory cannot be applied for plant networks, neither protein-protein interaction networks nor to the metabolic networks. So just to finish a little bit about metabolic networks, which is the aim of this class. So I hope now you have already a brief understanding of networks, and let's go to the metabolic network. Metabolism, as I said, can be constructed in terms of network as a network. So you have metabolites being the nodes and the enzymes being the links. So one enzyme take one metabolite, which is the substrate, to produce the product of this reaction. And here we have a metabolic path, not a really network, because metabolic network, you include all interactions that these metabolites can have with the enzyme. So this is a metabolic pathway, not network. Metabolic network is this one. So here you have a plant metabolic network. I showed it to you in the previous class. Basically, you have circles are nodes, and they are connected by links when you have interactions between the metabolites or the enzymes that produce these compounds. So you have also in green the metabolites, the reactions of the chloroplast network. In blue the cytosol, in red the mitochondria, orange is the peroxisome, pink I think it's vacuole, and black it's something that is getting from outside, for example oxygen, CO2, water, and so on, nutrients and so on, light. So you see that there's a huge connection. Everything is, not everything, but many things are connected to the cytosol. Like cytosol is a kind of buffer of plant cells. Okay, that's why it's very important. So it's a kind of it's a location where it's everything integrated, the chloroplast and mitochondria, peroxisome, and so on. Another important thing about metabolic networks is that you can transform every reaction into models. So for example, you can take a look on this system boundary, these reactions A, B, C, D, and E here. You have, for example, the compound A can go out of the cell or can be transformed to the B. The B can be transformed to the C or to the D. The D can go out of the cell, and the D can transform in E, which can also be produced from C. So every reaction here, you have a identification, for example, V1, V2, V3, V4, V5, V6, 7, and so on. And B1, B2, B3, B4, and B5. So remember that I said that you can measure the relationship of these interactions. Every interaction of the network, you can measure that. You can measure, for example, the enzyme activity or the correlation between these two metabolites or the correlation between two genes or two proteins and so on. So we have here, with this kind of information, we have this as we call flux balance analysis, FBA. So this is a mathematical framework to model metabolism. Okay, what this kind of model provide the metabolic flux for each reaction. So for example, the reaction around the metabolite A depends on V1 and B1. For example, the metabolite A can be transformed to B or can get out of the cell. So this is something around A. But the B is a little bit more complex because it can be produced from V1 and V4, but can be degraded by the V2 and V3. So the balance of these reactions, they usually put equal to 0. Okay, and then you have a matrix notation S versus V equal to 0. So there are many papers here that you can check a little bit more about flux FBA. It's a very nice modeling framework to create and also to interpret metabolic reactions. So what we can do with this information? So you have the two things, the experimental part about metabolism, and you have the computational part about modeling. The computational part is sometimes we call this as genome scale metabolic models, because you have all the information from the genome and then you use this information to create models that simulate metabolic response, even at subcellular compartment, like they will provide information about chloroplast, mitochondria, peroxisome, and so on. Okay, every reaction of the organism is gonna be predicted. And then you have the experimental part, which you do by doing metabolomics. And then if you have difference between what was predicted by the model part and what was measured, then you have this difference and accurate the model. So you fix the model. So then your model is gonna getting better all the time. And then you have metabolic flux distribution. Okay, so once you have the network created, the metabolic network created, you can predict anything, right? If you have the model ready and you have a good model, you can change, for example, remember that in the plant metabolic networks, you have some input like oxygen and light, CO2, water, nutrients, and so on. You can simulate the changes in the metabolic network by removing, for example, light, or reducing the amount of light, or increasing the amount of light, increasing the oxygen, increasing the CO2, and so on. Okay, you can play. Actually, you play. You choose one possibility, test, create the hypothesis, and then design the experiment and do the laboratory work. This is actually the strategy that they call building, testing, learning, designing cycle. Okay, this is using for engineering cellular metabolism of every organism. Okay, so you have models, you have, again, like I showed in the beginning, you have the experimental part, you have the modeling part, you have the hypotheses that have been created, and then you test and validate the model. So this is something that every field is doing: building, testing, learning, designing cycle. Every time we do this, we're rounding and improving our knowledge about the process that we are going to manipulate